a rising fifth-year graduate student at Rice University. Today, I'm going to share with you my findings in my dissertation project. Understanding the mechanisms that contribute to the evolution of reproductive isolation is a key issue in speciation. I'm particularly interested in how divergent selection interacts with gene flow to affect this process. There's a rising amount of evidence showing that there's higher reproductive isolation between populations under divergent environment than between populations under the same environment. Often with gene flow, ongoing gene flow between populations. Stressing the role of divergent selection in promoting the prezygotic reproductive isolation. However, we still are unclear about whether the presence of gene flow affect the prezygotic reproductive isolation. Gene flow could constrain the prezygotic isolation through homogenizing effect. It could also promote prezygotic isolation through reinforcement. There's limited empirical evidences assessing the interaction between gene flow and divergent selection in this process. However, understanding this question is important for us to improve our prediction in whether speciation occurred under divergent selection. So I aim to address this knowledge gap by using B-treaty host-specific gall-forming wasps that induce a plant root galls for its sexual generation and plant leaf galls for its asexual generation. Two host plant species feed on Crocus virginiana and Crocus geminata that geographically overlapped in southeastern United States, where Crocus virginiana trees extend its distribution to the west of Mississippi River. For the rest of my talk, I will use QV and QG to refer to the gall wasp populations that feed on each host species. In this system, we already know that populations feeding on two host species are under divergent selection due to both the phenological divergence between tree species and also local adaptation to host immune system. We also know that geography is a good approximation of the degree of gene flow or degree of genetic isolation based on both the mitochondrial DNA and genomic data, showing that B-treaty populations split genetically between allopatric and sympatric areas. Consistent with our prediction that sympatric populations experiencing higher gene flow than populations in allopatric areas. And what's more, studies in QV and QG populations in sympatric areas have found evidence of partial sexual isolation. As illustrated in this graph that a population frequency between individuals come from same host plant higher than population individuals come from different host plant. So I want to test the interactive effect of divergent selection and gene flow by extending this comparison to allopatric areas with less gene flow. And as a note that the degree of the differences between the same host plant and different host plant indicate the degree of the sexual selection. So I want to compare whether the degree of sexual selection increases or decreases in allopatric areas. Increase of the sexual isolation in allopatric areas, suggesting that gene flow constrained the evolution of sexual isolation. And this could also detect statistically by looking at host by geography interactions, significant interaction indicating the degree of the differences between same host and a different host changes once you move to different geography. And if I found there's a decreased sexual isolation in allopatric populations, that will suggest gene flow promotes sexual isolation, possibly through reinforcement. So to test this, I collect gall wasp populations across its geographic range, rear the males and females out, pair them in a non-choice mating trial. I observe their behavior every 2 minutes for 30 minutes in total, and write down their mating behavior, including male wing buzzing, male mounting, and copulation. So to have a general pattern, consistent with the previous finding, we found copulation frequency is lower between individuals come from different host plants. Once we extend this comparison to allopatric areas, the sexual isolation between different host plants remains. However, the degree of the differences is similar. Therefore, does this mean there's no effect of gene flow on the evolution of sexual isolation? Well, according to literatures, in the early stage of speciation, there's a lot of asymmetric sexual isolation going on, depending on whether which population origin you're looking at and which sex you're looking at. So I dive deeper into this data set by quantifying male preference, measuring whether the male wing buzz or not, and female preference by quantifying whether female copulate or not after male have displayed the commercial behavior. So I'm going to divide this mate preference by both sex and different host origin, and I'll go through the results one by one. So here, the mate preference in QV males. As you can see, in sympatric areas, it doesn't show assortative mating based on a female host plant. And in the allopatric areas, also there's no assortative mating based on host plant, even though you can see there's an increase of the overall male wing buzzing frequency in allopatric males. And for QV females, similarly, in sympatric areas, no assortative mating based on male host plant. However, in allopatric females, they mate significantly less with males come from different host plant. And the host by geography interactions also is marginally significant. So this suggesting that gene flow constrains the isolation. And then I'm going to show you the mate preference in QV populations. I just want to remind you that QV distributions fall in completely within the QV populations. So there's no allopatric QG populations. So to do the comparison, I compare whether QG individuals respond differently with females come from either sympatric areas or allopatric areas. And in QG males, we see that they didn't differ in their response to either females come from allopatric areas or sympatric areas, even though they have a strong assortative mating, but no signature of reinforcement. However, in QV females, we see that QV females mate significantly less with males come from the sympatric areas than allopatric areas. This increase of sexual isolation in sympatry are suggesting the involvement of reinforcement. To wrap it up, we found strong assortative mating based on a host plant in QG males, no assortative mating in QV males, regardless of the geology of the male population. We found assortative mating in QV and QG females, but depends on which geography you're looking at. So for QV females, assortative mating is higher in allopatric areas, suggesting gene flow constrains the evolution of sexual isolation. However, for QG females, assortative mating is higher in sympatric populations, suggesting reinforcement. And to further confirm the prediction of the reinforcement, which is lower hybrid than fitness, we went to do this hybrid process When by bagging males and females from either same or different host plant, and when the females oviposit to the leaves, they leave a permanent oviposition scar. Therefore, we were able to quantify the proportion of galls produced per egg and the fitness measurement of the wasps. And here on the y-axis is different cross types, male by female. And first, the results on the QV trees show that hybrid has significantly lower fitness than pure process on QV trees. Interestingly, the QG process on non-native host plant is even higher. And similar pattern is also found on the QG trees, where hybrid fitness are lower than pure process on native host plant. With that, I want to thank all my funding agencies and my lab mates, especially the American Society of Naturalists and Society of Study of Evolution to facilitate my field work. So I have extra time, so I'm going to talk a little bit more about I don't know if I have time to address the possible underlying mechanisms of the pattern we see. So just to review, we see this asymmetric reinforcement that QG females show reinforcement but not in QV females. And what's the possible reasons? So the strength of reinforcement determined by two factors. One is the strength of gene flow, the other is strength of the fitness cost of hybrids. As I showed before, there's no asymmetry in the fitness cost of hybrids. So we're looking at whether there's a symmetric pattern in the strength of the gene flow, which indicate that we're expecting to see there's more gene flow coming from QV to QG than QG to QV. And by looking at the morphology of the QV and QG wasps, we found that QG wasps have significantly smaller wings compared to body size, and the lab experiment testing the flight ability also showed that QG wasps cannot fly. So that indicating that QV individuals having stronger migration rates further confirms our hypothesis that there's asymmetric gene flow going between QV and QG, with QG experiencing more migrants from QG populations. And also, this also further confirmed why we see QG males showed assorted mating but not QV males. Because in general, QV populations experiencing more QV migrants, not the other way around.